Welcome back to day 15, chapter 16 podcast. In this podcast we're going to cover objective 2 shown here. And in this podcast we're going to describe the gene itself, the parts of the gene, the proteins that are necessary to bind to that gene in order to make the RNA molecule, and describe briefly some of the three stages of transcription: initiation, elongation, and termination. All right, so let's go ahead and get started here. All right, to get us started here, let's just go ahead and draw a basic gene, your favorite gene here. And we're going to put our 5' in, and then 5' down here, and 3'. As we saw in the last podcast, this is our template. And up here is our non-template. I'm just going to put NT for non-template. Let's draw some basic components of it. We know that if we were to draw kind of like an imaginary line here, that this first region here would be called our promoter. I'm going to draw it up here on top, but recognize that this whole region would be called the promoter. And this main part, the big part of the gene here, is called the transcription unit. Transcription, my shorthand for transcription, TXN, and unit. This is the part that codes for the protein. And at this very far end, it's our termination sequence. So I'm going to put TER, termination sequence. Sorry, I ran out of room there and it looks sloppier than usual. The promoter can have several different kinds of nucleotides, but there are some sequences that have been conserved between almost all genes that we know of. And the first one, it's a very important one, it's called TAT. It's known as the Tata box. This is a very important part of the protein. And we'll talk, of the promoter. And we'll talk about that in a moment. The transcription unit will always begin on the template strand. It'll have a TAC. And if that doesn't make sense, think about when this template is made into an RNA. It's going to make an AUG, which is always that first amino acid codon in the gene that will make the protein with methionine at the front. And then we know the last 3 bases in this transcription unit will be a stop codon. And if you look at the genetic code, there are 3 possibilities. One of them is ACT. And when this is made into a messenger RNA, then this will be a UGA, which is a stop codon. The terminating sequence here is various sequences. Don't worry about the sequences in the terminating sequence for right now. Maybe when you get to genetics or cell biology, you'll get into that a little bit more in more detail. But you should know that the sequence is such that when the machinery moves this way, making the RNA, when it sees this terminating sequence, it stops the transcription of the gene. Okay, now transcription has 3 main parts. The first part is initiation. The second part is elongation. And then the last part is termination. Let's begin with initiation. And let's begin with a protein, an enzyme called RNA polymerase. And I'm going to draw it as this big green circle, and it binds to the promoter. More specifically, it identifies the Tata box and binds to it. The promoter, well, let's take a step back. Remember when we talked about DNA polymerase and replicating DNA, that we had to have all these proteins. We had to have a helicase to unwind the DNA. We had to have the topoisomerase so there wasn't that torsional stress on it that would break it. And we needed a primase to make a primer. RNA polymerase is such that you don't need any of those things. RNA polymerase will still read 3' to 5' to make an RNA molecule. It doesn't require a separate helicase in theory, but it does actually have a component of the RNA polymerase that acts as a helicase that will rip those hydrogen bonds apart. And a very advantageous aspect of the RNA polymerase is that it does not require a primer. It can just start making RNA molecules. And so let's see. I'm going to write the word RNA polymerase here in green so you can match it up with the green up there. RNA polymerase. Now, if this was a prokaryotic cell, that's essentially all you would need to have transcription initiate. In eukaryotes, so let's make a different color. In eukaryotes, we would have something called transcription factors. So I'm going to write TXN for transcription and then factors. And in parentheses, I'm going to put EUKS for euks. These are only found in eukaryotes. And in order to regulate this gene, which we'll talk about gene regulation later, somewhere between 20 and 50 different specialty proteins need to bind to either the promoter or to the RNA polymerase itself in order to kickstart this RNA polymerase so it begins to move. So these are transcription factors, incredibly important. They're linked to many important cellular processes, physiological processes like development. They're also linked to pathology. Many cancers are linked to these transcription factors as well. There's many transcription factors. Okay, so that's all we really need to know about initiation. Now, elongation, what happens with elongation, and let's pick a different color. As elongation continues or moves from this promoter and hits this transcription unit of the gene, it will begin to make an RNA molecule. It might not be the best way to draw it, but I'm going to draw it like this. So this is as it's elongating and it's moving down this transcription unit here. It's making an RNA, messenger RNA molecule. As it's moving down here, that helicase that's a part of the RNA polymerase will rip those hydrogen bonds apart to make the RNA molecule. This moves at a pretty fast pace until it hits this termination, or the terminator sequence. Once it hits this terminator sequence, a few things will happen. The RNA polymerase will fall off. I'm going to draw that here. It's kicked off, and it goes on to transcribe another gene, but it's off of this gene for now. If it's a eukaryotic cell, all of the transcription factors will get kicked off. It's not really that important for this class right now, but recognize that these transcription factors, some of them will come off at various stages of this process, but certainly by termination, they have all been released. And perhaps the most important thing that happens is that our newly made RNA molecule is released. Remember, it's anti-parallel with the template strand, so it's going to be 5' to 3'. And then once it's released, in eukaryotes, it's taken into the cytoplasm, so it can be translated. Remember, if it's bacteria, it just stays in the cytoplasm because this all occurs in the cytoplasm in bacteria. Now that, for the most part, ends this objective. And we'll pick this up for next class when we start talking about how this messenger RNA here, this messenger RNA, before it can be translated into a protein, what has to happen to it. Particularly, we're going to talk about what has to happen to it in eukaryotes. Now, before you log off here, I want to show you an animation that's part of your learning material with the mastering. It's one of the BioFlix videos that they have. So let's go ahead and show that and talk a little bit over and see what they're doing. Okay, this is one of the animations that the publisher provides, and you have access to these as well. But it begins with this short little story here of this student cramming for her biology exam, and she's eating some sugar, and then eating this sugar, we know that an enzyme called insulin is produced, and this insulin is important in regulating the sugar. Now what they're going to do is take a closer look here and go inside the cell and ask, how is that enzyme ultimately produced so that it can do its job? And so here's our tour inside the cell. You see some familiar features, but ultimately we want to get inside this nucleus and see what's going on in there. There it is. All right, so let's review a few things here. Here's our DNA. These are the histone proteins that wrap the DNA up into a nucleosome. Now remember, the DNA is tightly compacted, but every now and then there's a gene somewhere along here that we need access to so that we can make a protein. So here's an RNA polymerase, and as you see here, it's moving along here. Now this particular video doesn't show the transcription factors, but remember there are transcription factors also bound to different parts of this that aid in the transcription. But remember, as I said, the RNA polymerase will bind to the DNA, rip open those hydrogen bonds to start making the RNA molecule. And that's exactly what we see here. Here's the RNA polymerase, and if we look inside the RNA polymerase, we see that it is, as it's ripping it apart, the double helix, it's adding the complementary base to it. As it's adding the complementary base to it, it's kicking the RNA molecule out. Okay. The RNA, you'll see, I'm going to hit play in a second here, it's going to keep moving in this direction. As it makes the RNA, you'll see it's going to continue to reform that double helix once the RNA is produced. See how the helix is being reproduced here? It's unwinding here. We're adding the bases. Again, it's following the complementary pattern. Okay. We're going to stop it here, but we're going to come back in the next podcast and talk about processing of the RNA molecule. All right, well, good luck, and if you have any questions, please let me know. I'll see you later. Bye.